This is the placenta. It is a transient, 500-gram, disc-shaped fetal-maternal organ that serves as the primary biological connection between mother and fetus. Its physical position is typically high within the upper uterine segment, where it anchors firmly to the wall of the uterus. From this position, it must coordinate two entirely separate circulatory systems belonging to two genetically distinct individuals. The volume handled at this junction is immense. Each minute, 900 milliliters of blood flow into this single organ. Despite this proximity, these two distinct fluid systems cannot be allowed to mix. If they did, it would trigger a lethal immunological reaction from the mother's immune system. To maintain the pregnancy, the organ must facilitate a high-volume exchange of life-saving gases and nutrients, while ensuring that the physical barrier remains absolute. To understand how this works, we have to look at how the organ is built, layer by layer, from the uterine wall inward. What we find is a specialized anatomical structure designed to manage the high-stakes interaction between these two separate systems. We can begin the reconstruction with an empty space, the gap between the mother's uterine tissue and the developing fetus. The first required boundary is the maternal surface. This side is formed by the mother's own tissue, specifically the decidua basalis, or decidual plate of the endometrium. This maternal surface is rough and irregular, divided into segments called maternal cotyledons. To complete the enclosure, we need a second boundary, the fetal surface. This second plate is contributed by the chorion frondosum, which forms the chorionic plate. Unlike the maternal side, this surface is entirely smooth. It is covered by the amnion and serves as the attachment point for the umbilical cord. When fully developed, this dual-plate structure reaches roughly 15 to 20 centimeters in diameter and 3 centimeters in thickness. By locking fetal tissue against maternal tissue, the organ creates a sealed internal chamber where the actual work of exchange can take place. We now move inside the gap trapped between the basal plate and the chorionic plate. This specific void is known as the Intervillous Space, and it has a maximum volume of 140 milliliters. The mother's circulatory system floods this space, pumping maternal blood into the void at 500 milliliters per minute. To reach this maternal blood, the fetal side projects 40 to 60 structural extensions, called stem villi, down from the chorionic plate into the void. These stem villi divide into a complex series of branches and terminal ramifications. These finger-like structures are the chorionic villi. Some of these, known as anchoring villi, reach all the way across the space to physically embed in the decidua basalis, acting as structural pillars. Others, the floating villi, do not touch the bottom plate. They remain suspended within the pool of maternal blood to maximize contact. Fetal blood flows strictly inside these closed villi at 400 milliliters per minute, allowing it to pass near the maternal pool without ever mixing with it. This design increases the surface area for contact between the two systems without requiring a direct connection between the blood vessels. The internal architecture functions like a submerged forest, where sealed fetal roots soak up resources from a rapidly flowing maternal river. If we zoom in from the macro structure of the villi down to the cellular level, we find the actual point of contact. This is the final barrier between maternal and fetal blood, an outer layer called the syncytiotrophoblaст. This membrane is highly specialized for transport. It thins from 0.025 millimeters early in pregnancy to just 0.002 millimeters by the later stages. While it is thinner than a sheet of paper, the total functional surface area of this membrane expands to cover up to 14 square meters. This microscopic layer is responsible for performing 6 distinct physiological roles simultaneously. First, it manages transport. It allows for the passage of oxygen, water, and nutrients like carbohydrates and amino acids. The fetus takes up about 25 milliliters of oxygen per minute through this process. Second is elimination. The barrier pushes fetal waste products, including carbon dioxide and urea, out into the maternal blood. Third is immunity. The membrane pulls maternal antibodies, specifically immunoglobulin G, across to the fetus to provide protection against infections like measles. Fourth, it acts as a barrier, physically blocking bacteria and other harmful substances from reaching the fetus. Fifth is its synthetic function. The layer produces hormones like progesterone, which is required to maintain the pregnancy after the 4th month. Finally, it manages exchange, carefully regulating the interaction to keep the bloodstreams separate and prevent a lethal reaction between them. By managing these 6 roles, the syncytiotrophoblasts performs the work of the heart, lungs, kidneys, liver, and immune system for the fetus. We must now pull back from the microscopic cellular barrier to observe the complete exterior view of the organ. While the standard model is a single disc, there are often variations in how the organ is structured. As listed here, a placenta can develop into different shapes, including bidiscoidal, oval, triangular, or irregularly lobed forms. The umbilical cord, the primary fetal lifeline, also features different attachment patterns. These include marginal attachments at the edge of the disc or velamentous insertions, where the vessels travel through the membranes before reaching the placenta. Even the distribution of the umbilical arteries varies, categorized as disperse, magistral, or furcate types. These variations are more than just anatomical details. They can influence how efficiently the organ manages blood flow and supports the pregnancy. The anatomical evidence shows that the placenta is a complex, highly specialized, and transient multi-organ. Its intricate cellular architecture and precise control over fluid dynamics are what make mammalian gestation physically possible.